Recently, Roctavian was in news, which is a gene therapy to cure hemophilia A. Hemophilia is a rare hereditary condition which is due to defective gene on X chromosome of human beings, and due to this defect there is a decreased synthesis of certain clotting factors. Clotting factors are those proteins which are required for clotting of blood following any injury. In the absence of clotting factors, there will be continuous bleeding following a minor injury. Sometimes there may be spontaneous bleeding in different internal organs of the body. So mainly hemophilia affects males because in males there is only one X chromosome, whereas in females there are two X chromosomes. So if that single X chromosome is affected or mutated, the males will be affected and the disease will be manifested. Hemophilia is of two types, A and B. In hemophilia A, there is a deficiency of factor 8, and in hemophilia B, there is a deficiency of clotting factor 9. Hemophilia A is far more common than hemophilia B. So there are different modalities of treatment. Replacement therapy is a conventional treatment. Here the concentrates of clotting factors are injected into patient's body through the vein. So factor 8 is injected for hemophilia A and factor 9 for hemophilia B. The replacement therapy has certain disadvantages because the concentrates derived from human blood, it should be tested and screened, otherwise the blood-borne diseases like hepatitis can be transmitted to the person who will receive the concentrate of the clotting factors. Hemophilia patients regularly use this replacement therapy to prevent bleeding, and it is meant to protect against unexpected bleeding episodes. So there are many challenges in this replacement therapy because one of the major challenges is body's own antibodies can sometimes destroy the clotting factor before it has a chance to work. So the whole idea of replacement therapy is defeated if the antibodies work against the clotting factor. Second modality is desmopressin therapy. Desmopressin is a man-made hormone which is used to treat people who are mild hemophilia A. It is not used to treat hemophilia B or severe hemophilia A. So this hormone increases the level of clotting factors in the blood, so helps in preventing bleeding episodes. Gene therapy is a new and emerging approach. Here copies of corrected genes are introduced into the cells of a patient. The idea is that there will be a normal expression of necessary clotting factors. Roctavian is the brand name which is the first and only approved US Food and Drug Administration approved gene therapy for hemophilia, and this is an adeno associated virus based gene therapy, and it is approved only in 2023 for treating adults with severe hemophilia A. It is only for those without any pre-existing antibodies to associated virus or adenovirus related viruses. It is a viral vector carrying necessary gene for clotting factor 8. The gene is expressed in liver to increase blood levels of factor 8 and reduce the risk of uncontrolled bleeding in patients. The effectiveness of Roctavian is established based on results from 112 patients who were followed up at least 3 years after Roctavian treatment. Majority of the patients who received this gene therapy got corticosteroids to suppress the immune system for the gene therapy to be effective and safe, because treatment response may decrease over time. The Indian trial took place at Vellore. The main difference is the vector. Here lentivirus is used instead of adenovirus. And the advantage is that adenovirus infections are far more common in people, so the chances of antibodies are fairly high. So this can be counterproductive to those receiving Roctavian, whereas the Indian based gene therapy, it includes lentivirus and the infections are less common than adenovirus, so the chances of antibodies against this lentivirus is very less. So it is expected that this lentivirus based gene therapy is more effective than Roctavian. Further, the Indian approach relies on gene transfer into the adult stem cells with lentivirus vector that integrates the body's cells instead of in vivo transfer to a hepatocyte or a liver cell. So through a non-integrated AAV virus. So Indian gene therapy is more reliable. It produces lifelong clotting factor in necessary quantities without any side effects. So these are the advantages of Indian gene therapy over the Roctavian. So only this Indian gene therapy was tested in 5 patients, so it is not more reliable. It is on a trial stage, but none of them reported bleeding episodes in the trial.